share my screen?
Yeah, yeah. Yes, you can start and you can switch off camera if camera if you switch on it might consume more internet. It may be breaking down, so you can switch off. That's fine.
Okay,
Up to you.
all right.
Mm.
Yeah,
Mm.
yeah, yeah, yeah. Then, yep. Is it visible? My screen is
Yes.
visible.
Yeah.
Yep. Yeah.
Screen, screen is visible and
Firstly,
voice also clear from my end, and others also hope it is clear to everyone. Hi, Manshu, it
yeah.
is clear from everyone.
Yes, yes, it is clear.
Okay. Okay. Yes,
Thank
Partha, you
you.
can carry on.
I'm
Yeah.
glad. Well, let me introduce myself. My name is Pardhasaradhi Mathi. I'm presently working as a senior development scientist in R&D, process development and technology transfer for NHS BT in UK at Bristol side. Well, I'm glad for overall understanding of the regulatory aspects and the basics of regulatory from the Fortunate Veda group. Especially, I would like to thank Mr. Reddy Garu for providing an overview of complete training session for the whole 3 months. So, as an assessment of my course, I'll be presenting today an overview of regulatory affairs and terms, basics for the advanced therapeutic medicinal products in aspects of gene therapy, especially. So, moving forward, I'll be my agenda, whichever I'm going to cover today is overview of ATMPs and mainly other terminologies in regulatory affairs, which we'll be using daily in terms of regulatory norms. And the third one is the ICH guidelines, which I learned especially in this course in duration. And finally, the dossier structures, the CTT structures, whatever we'll be using, especially for the gene therapies. Moving forward, as an overview of ATMPs, what are ATMPs? Why it is termed as ATMPs? Advanced therapeutics medicinal products, especially these therapies are evaluating in terms of evolution. Compared with other therapies, these therapies are more in depth or more personalized towards the therapy, towards the disease. Hence, that is more advanced. In those aspects, it is termed to be as advanced therapy medicinal products. And the main reason of presenting here of regulatory is a more gray area, I feel. A more limited knowledge and the scales set is available in the market. Hence, that's the reason I prefer to learn this course, especially on the gene therapy aspects. And CAR-T is one of the another domain of ATMPs. Stem cell therapies is another vast area of ATMPs, and more budding areas, advanced antibody drug conjugates. And meanwhile, where I'm working on various products on these areas, especially gene therapy products on adeno-associated viral vectors, CAR T therapy related vectors, hematopoietic stem cells, and so the main work which I'm doing at the moment is on adeno-associated viral vectors and lentiviral vectors. So hence, um, I'll be more focusing on these areas of the basic of understanding to the presenters, to the to the listeners. So I will be focusing on the area of recombinant adeno-associated virus production as well as the lenti production. So the main in this slide, the first thing is the recombinant adeno-associated virus production process. In this process, there will be the raw material is a three plasmid resource. In this, the gene of interest is the first plasmid, obviously. The second plasmid is a rep and cap plasmid, which includes the structural genes in that. And the final most is the helper plasmid. In this, the process includes using these three plasmids. We will be doing a transfection with the polymer, which in general we'll be using a polymer using a decorated cell line, which is HEK293 cell line, human embryonic kidney cell line. Using these three plasmids along with club merged with the polymer, we'll be transfecting the cells, and upon receiving by the clathrin receptors, the cells immediately breaks the polymer inside the cell, with the aberration in the pH. Then the plasmids undergo inside the nucleus. Then the replication happens. Then the post-translational modification. In the post-translational modification, the plasmid, the virus accumulates, starts to produce there, and packaging of these gene of interest happen inside the virus. In adeno-associated virus, there are various types from AAV1 to AAV9 different serotypes, and the gene of interest merges inside the virus. Hence, this is another critical thing in the following presentation I'll explain you. And purification is a very challenging aspect in this. And coming to the second process I'll explain is the lentivirus. The main difference between those processes is usage of the four different plasmids compared with the AAV. In AAV, we'll be using three plasmids. In the lenti, we'll be using four. The first one is the gene of interest, as usual. Second one is the GAC-Paul structural plasmids. Third one is the Rev plasmid, which is another functional plasmid. And the final one is the envelope plasmid, that is VSVG. VSVG is usually comes from the most of the viruses, especially linked to the HIV as well. So again, the process comes again the similar way. The four plasmids will be again transferred to using a polymer, but again coming to another cell line, that is HEK293, but containing a typical SV40 antigen, then the term as T cell. So here the similarly the cells will uptake the polymer with the plasmid. Then the rupture of the polymer departures the plasmids to the nucleus. Then the packaging happens inside the cell with the in the cytoplasm. Then the main difference with the AAVs. Okay.
Yeah. But
Yeah. The main difference between the AAVs and lenti are lentis are
Pardhasaradhi,
completely.
just sorry to
Yes.
interrupt.
Yeah.
Can we take questions in between or at the end of the session?
Whichever you feel, I don't mind.
Sir, is there any question, sir?
before going to next slide, we'll ask. Okay.
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 Yeah. Yeah.
So yeah, in especially in the lenti process, you will see only the budding virus. This is only one serotype. This is only budding virus. The cell bursts out and produces the virus outside the supernatant. So yeah, before going to the next slide, I want to take the question. What who wanna ask me?
So, Partha, these four plasmids are there, na? They will be integrated into a single plasmid kind of thing?
No, no, all four are independent.
Independent plasmids.
Yes, all four are independent with specific elements containing in that, and
Yeah, yeah,
each
yeah.
plasmid have their own specific nature to produce a virus.
Mm-hmm. So all these are integrated into the genome.
No, no, no.
Okay.
In adeno-associated virus, no, none of the plasmid will get integrated into the HEK genome. In
Okay.
lenti, probably yes.
Mm-hmm.
When we treat the patients, adeno-associated virus doesn't integrate into the human genomes. In lenti, it is a raw material. In lenti case, lenti acts as a raw material.
Okay. So how do we make sure that all these, I mean, like these four plasmids
Mm,
are important?
mm,
In a way, some of them are useful in the packaging, in
mhm.
envelope or something kind of
Yes,
way. So
yes.
how do you make sure that the lentiviral particles that are coming out will be having these four plasmids are like,
So in a release criteria, we have
okay,
to
okay.
ensure that in the release
Mm-hmm.
criteria.
Okay.
Yeah. Your name?
Narasimha.
Seema. Okay, thank you. Any
Yeah.
more questions from the participant? Okay, I'll move forward then. So in both the cases, in both the situations of adeno-associated virus as well as the lentivirus, both includes plasmids. Here the plasmids is a initial raw material for the process. So in the process of plasmid generations, we'll be using E. coli as a master cell bank in the strain production, and we'll be generating a seed. From the seed, we'll be producing a fermenter. With the help of fermenter, we'll be generating the production of the E. coli. And in the E. coli, the plasmid will be generating. And to produce the plasmid with the help of alkaline lysis, we break down the cell wall, then the plasmid comes out. In the plasmid generation process, it is very sensitive process at the time of harvest, and we'll be using a various alkaline lysis steps and subsequent wash steps with the PBS. We'll be further doing a pre-purification steps. Upon completion of the pre-purification, we'll be using a TFF. With the TFF, we'll be concentrating the material. In the TFF, we'll be concentrating the material, and further we'll be loading the material to the chromatography. There we'll be removing most of the mRNAs or residual materials of host cell DNAs or guide RNAs, guide DNAs, endotoxin removal. Further, we'll be using most of the ultrafiltrations to remove the unnecessary impurities. Here, the raw material is very, very, very critical to produce a good quality of virus. Because of the unnecessary impurities, the transfection will get hampered. Moving forward to the next step, the next step is the viral production. Hence, as I said in the previous steps, in the previous slides, the plasmids will be used for the HEK cells. In the HEK cells, we'll be preparing a different banks. As usual, we'll be criteria of selection is based on the again ICH Q4. Again, these cells will be used for the expansion in the bioreactor. In the bioreactor cases, we'll be expanding and doing the transfection. And subsequent step within in the case of AAV, we'll be using the post transfection 72 hours as a harvest. And in case of lenti, we'll be using a 48 hours of post transfection as a criteria for harvest. And then in the case of AAV, we'll be using a lysis step. And in case of lenti, we'll be using just straight away a benzoate step. In case of AAV, in few serotypes, especially AAV5, AAV2, AAV8, we will be using a lysis to break down the HEK cells using a detergent. And benzoate treatment, upon benzoate treatment, we will be using a benzoate and endonuclease as well. In the benzoate treatment, we will be making the unnecessary plasmids or host cell DNAs, host cell mRNAs will be chopped off in this with a 1 hour treatment at 37 degrees centigrade. And one of the critical step in the process is the clarification, depth filtration. Sometimes it gets choked based on the polymer we use. And further final downstream steps starts from this ultra TFF chromatography steps. Upstream process hardly covers by 10 days in the manufacturing stages, in the 10 days, and another 5 days will be doing in the downstream process steps. Downstream process steps starts with the capturing steps. In the case of lenti and the case of AAV, we'll be using a capture. In the capture, most of the full capsids will be captured, and few of the empties will be attached to the ligand of the resin. And further subsequent washes with the various buffers will be making the empty capsids will be flow through in the flow through, and full capsids will be collected in the next end of the flow through. And further steps, again we follow the TFF concentration, and dye filtration happens, and sterile filtration will be processed. In the final product formulation, we especially use sucrose for the stability of the viral vectors, and we also use glycerol along with the pluronic F68 to maintain the minus 80 degrees centigrade stabilities. Moving forward to the another slide. Yes, as per one of the participant question, these are all the release criteria we'll be following. Same
So,
in the left side
Prakash,
of the yeah
excuse me.
yeah.
Before going further, so
Mm,
this benzonase treatment you are
mm,
doing,
mm.
so in the here,
Yes,
so this
yes,
won't affect the virus particles. So it
no,
will be specifically killing or helping in
yes,
removing the HCD and the plasmid DNA.
yes, yes, correct.
So this virus is of DNA based, no? Whatever we are getting virus particles here. Or if,
inside the virus, yes, our gene of interest will be there. Correct.
okay, okay, the virus is protected by that capsid. That is why
Yes.
benzoin
Correct.
is
Yes, Benjones won't be acting on the inside the gene of interest. It is specific to the impurities available in supernatant,
okay.
especially the host cell DNAs, host cell RNAs,
Mm-hmm.
mRNAs.
Mm-hmm. No, my question is like benzoin is just a chemical. It is not specifically targeting some kind of DNA or thing kind of. It is
Mm.
like kind of. So how it is protecting? Or is it
So
protecting?
yeah, it is having a capsid, hence it is protected. And other step is we take care of the temperature of the bioreactor at the time of Benjones treatment, and the
Okay.
time is also very critical. And at the time of process development, we have to optimize at a specific concentration of this Benjones treatment. Specific units per ml is essential based on the cell count at the time of harvest.
Cell count in the sense whatever the particles are coming now
Yes,
or
at the
virus
time of
particles.
cell count, what is the cell count in the supernatant in the cell broth? Based on the cell count, we have to optimize the concentration of these benzoate treatment, such as 10 units per ml or 5 units per ml, along with the magnesium chloride concentration, and
Mm-hmm.
specifically at specific time and specific temperature. We treat mostly with 45 minutes to 1 hour,
Okay.
and we have optimized that.
Okay.
So beyond that, yes, you can, but again, we will be adding a supplementation of EDTA to stop the benjones treatment.
Exactly. Exactly. Exactly.
Yeah.
Okay. Yeah. Okay. Fine. So Bharta, before going again, what is
Mm.
this HAK 293 T? T for what?
T is specifically we use for the lenti process. T is the subunit antigen. It is termed to be a tumor antigen. It is SV40 antigen, which
Okay.
will especially prolong the virus for LNT virus.
Okay. So you mean
Uh,
to say that the HAK 293 is genetically modified having
yes,
SV40. Is that
yes,
correct?
correct, correct.
Okay. Okay. Thank you.
Mm.
Thank you.
No worries, no worries. Yeah, shall I move forward then?
Yeah, so I would like to ask one thing here. Like these processes are where we are producing the viral particles. So would you like to shed some light the safety point of things? Like how is it all things are done in BSL level 4
Good
or how
question.
it is
Very
done?
good. Absolutely. I take this question. Yeah, especially we follow BSA biosafety limit class A, especially when we are purify in the bioprocess area. Completely in the bioprocess area, we follow the class A environment. Most of the things happen under the biosafety cabinet or the sealing labs. Most of the thing happen in the manufacturing, but in case of R&D, we were limited to use on certain stages in the biosafety cabinets or in the class A environments. So yeah, in the case of manufacturing, yes, most of the cases are class A's.
So currently, like because I believe that these are very much
Mm.
infectious in nature. Uh,
Mm.
right?
Y- yeah. Mm.
So it should be restricted to I don't know like what is the nature of the like
Mm.
the gene here, how it is
Mm.
infectious, where whether
Mm.
we are eliminating the infectious portion from the virus here. If we are eliminating, then I think that we can allow to do the things in R&D level, even manufacturing level also we can do. If not, then
Mm.
then it must be done under the BSL level, or as you said, the class 1 level kind of
Mm.
facility, even in R&D sector,
Yeah.
right?
Correct.
Yeah,
Yeah.
ideal
Correct.
situations, in the ideal situations, yes, we take care of the virus. Very ideal situations, we have faced a few issues as well in our lab, in our case. But yeah, in we have to be very cautiously usage of the viruses in the harvest stage as well, in the purification stage as well. Many of the times it will there will be spillage. Definitely there will be a spillage over the glouses. So we use always a sterile gloves until the handcuffs. Full sterile gloves we'll be using, and any sort of spillage we'll be immediately treating with the bleach, whatever the hypo solution. We have a specific capsules to treat the whatever spillages we have. And whenever we use the materials, even the TFF we use, even the bioreactor we use, everything is single use. Sometimes we use a TFF which is of multi-use, so we want to do the process again. So we do not want to dispose them, so we again treat the material. In some materials, so some pressure sensors in the downstream aspects cannot be treated with the, so with the bleach. So those we ensure not to touch with the contaminants, or not to touch with the virus situations, virus materials at all. So we ensure always, whenever we use any tapes, any sort of PCR we do, any flow cytometry material we use, any consumables, we always ditch them in the bleach material.
So,
So it,
I would also like to know one more thing. Like,
mm,
what is the scale of bioreactor we are operating in this kind of technical technology?
yeah, in case of AVs, or in the case of lenti, the process is similar, quite similar, but the material usage will be completely different. In case of AVs, you will be needing a more material. So hence people may go until even 200 liter scale of bio in the case of manufacturing in the pilot scale. In terms of lentivirus, there will be very less usage for the very mild, one or two batches should be enough. Hence, that's the reason people go maximum of 50-liter scale bioreactor scale at the manufacturing scale, and in the R&D people limit to the 50-liter scale for the lenti process.
Okay. Okay. In my side also, I'll add a few things to clarify the Himanshu questions. Yeah,
Mhm.
definitely the class air is required to handle that, especially the cell culture, because nothing Vachike 293 is equal to CHO cells, because this is also mammalian cell culture. So required to handle the upstream process to culture the cells, required class A area and sterile operations, close operations. When the virus infection is there, stage. So second stage, where the mentioned bioreactor expansion and transfection.
Mm,
This transfection stage is very harmful to the human and environment at the particular area. So required
mm,
biosafety containments like level 3, level 4. So which is the control environment where we can minimize the exposure of going out. The virus particle anything is flying in the area that should not go outside. So we should have the biosafety containments where can be within the area, so that within the area, if anything is there batch to batch, we can perform any fumigation kind of things. So what are the disinfectant procedures are there that can be used, so that we can minimize the particles can go out from the particular area
Hmm.
to outside of
correct.
the
Hmm.
area.
True.
And the persons peoples are entry exit
वो तो करो
having
मेरे को
overgoing procedures,
एसिडिटी गोली दो
the people having overgoing procedures, and people has to use single use garments, so that the person is entering into inside will go with sterile garments, will come out with our exiting during exiting. So you will remove the sterile garments, so that will be avoid any manual interruptions to cause any cross contaminations.
Correct.
Clear emotion?
Yes, yes. So these are the, as you said, that these are the exact requirement from regulatory end also. Whether it
Yes,
is in,
because
Yes.
first
Yes, it is.
agency itself, if you're not going to show this type of control, agency not allowed to perform this type of activities in our
Correct.
laboratory.
True.
yes,
True.
So
We
and
have to
agency will,
submit an
yeah,
initial
discussment then,
documentation that we have
yeah.
to.
and
Uh,
one more thing, Pardhasaradhi, can you go back to your slide? I just want to see the vector designing slide which you have just now shown. Can you go there back?
this one.
Yeah, yeah.
Yeah.
This one or back also you can go there. This.
Yeah. These are for the AAV.
Yeah, so these are for recombinant AAV. So here
Yes.
the structure is like that. We are having total three number of vector wherein
Yes.
our desired gene has been incorporated.
Mm. Yeah.
So I believe that some of them are producing the capsid, some of them are producing genetic material, or somehow
Mm.
everything will be integrated and then AAV will finally be produced.
Correct.
So, in this composition of genetic material, are we having the infectious gene incorporated, or we are removing those things, the
Yeah.
infectious
Yeah.
genetic material?
A good question. We will be making the plasmids with the devoid of all the infectious elements. will remove the infectious elements related in the plasmids. In a way, we will say that the plasmids are free from CAG elements, in a way. So most of the infectious elements will be removed
So
in
we
all
can
that.
say to the regulatory agency that here is one thing that it is we are not
Mhm. Yeah, yeah. See, while the submission of the CTD
that here
structure,
we are taking care of those.
yeah, we have to take care of these elements, especially at the time of submission of the CTD
Your
as well.
voice is breaking.
In the submission of CTD, we have to mention our raw material usages and what all the raw material sequences have, how the sequence structure is, and especially the all the sizes of the plasmids we have to mention. So it's a it's an open section. Any question on
Okay,
this?
okay, you can go ahead.
Yeah. So moving
కార్తీక్ అని
forward,
కౌన్సిల్ ఉంది కన్నా యాక్చువల్ ఫిబ్రవరి
yeah, coming to the plasmid release criteria. In the release criteria, as usual, everybody we have to mention the identity, purity, quantity, potency, impurity as well, especially in the release criteria of plasmid in the CGMP environment. In the release criteria, ITR integrity assay will be performing on the plasmids, UV detection, and the plasmids should be always supercoiled. So it is SC denotes the supercoiled plasmid that will be mentioned in two different methods, establishing in two different methods, HPLC as well as gel electrophoresis. Potency will be performed once in a time, that is functionality assay. Impurities, endotoxin, sterility, mycoplasma are the critical things to pass on to the GMP environment. And release criteria of virus particles in the right side of the panel shows AV capsid integrity, AV identification, whether this is of which serotypes in the Western blotting and sequencing. And the quantity is always measured in the dPCR. Nowadays, we use dPCR rather than the qPCR. And purity is always measured for the in aspects of viral vectors as empty versus full capsids. In the empty capsids or partial capsids, we see no integration of the gene. I mean, there will be any gene of interest in the capsid, and there will be partial amount of gene of interest inside the capsid, and the full capsid, which is of our product of interest, which is having a full amount of gene of interest in the full capsid. And we always mention in the upstream end, depend on the serotype, it varies from 5 percentage to the 30 percentage of full capsid in the upstream end. And in the downstream release criteria, we always ensure to reach to the 90 percentage of full capsids. And the final is the potency TCID 50. And these are the impurities analysis. Coming to the residual plasmid DNA, benzoate assessment, mycoplasma, sterility, and safety aspects, replication competent assay is very essential part as well in the release criteria, where the virus should not get integrated into the genome of the humans, into the patients. So these are the release criterias I want to, and these things are very critical in the submission of regulatory CTDs in the module 3. And this is also another criteria which I've seen recently in one of the publication, where they have to see the ITR integrity aspects into the replication. Cassette size, as I said in the AAV, we have to use only 4.7 kb of gene of interest, and in case of lenti we'll be using until 10 kb of gene of interest. And these are the identity of DNAs, identities, purities. Endotoxin bioburden will be used in the toxicology and GMPs. And residual host cell DNA, host cell protein, host cell RNA as well. pH in the case of this is final final formulated viral particle release criteria, tabulated structured one. So moving forward, as I said, since I have, after attending the course, I have you understood few terminologies of regulatory affairs, especially biological license agreement application, where we'll be using for all our biological substances. BLA, in the BLA we'll be using for the gene therapy, whether it is vaccination or biosimilars, ensuring the potency, purity, safety aspects will be mentioned in the BLA application. And new drug application is especially used for the small molecule structures, so especially the pharma molecules, so to ensure the safety and effectiveness. And investigational new drugs, IND, is especially for the clinical trial material movement over to the different materials, different stations. And abbreviated new drug application will be using especially for the new material for the marketing approvals. So these are the structures BLA or NDR IND will be using in the ECTD format always.
So these
So
terminology are based on US FDA framework, right?
yes, correct. In keeping of me with overview of US FDA, I have started presenting this.
Okay. And the similar structure is being followed in EMA also, or do they have any framework
Mostly,
for this,
yes.
for
EMA
this
as
kind
well
of
have
drug?
this. Yes, EMA as well having the similar kind of structure. Even EMA has even more stringent lines in the ATMP structure.
So in EMA, in addition to that EMA, instead of BLA, they will call MAA, Marketing Authorization
I mean,
Application. That is the
marker
only
and correct.
difference. A new
Mm,
drug application will be remains same, and initial new drug also remains same.
mm,
That what should be using for new drug application, using for the chemical drugs, generics. IND
mm,
is using for initial new drugs. That is common terminology in all
mm,
ICH. So
mm.
only the change is BLA versus marketing authorization is the differentiation. But the
Yes,
the requirement is same. The intended purpose is same.
correct,
Okay.
correct. Moving forward, yes, after entering the core structure, I understood about the ICH guidelines, especially four different guidelines in the market available over the ICH website portal. Especially ICHQ terms the quality having 26 guidelines, safety terming 16 guidelines, efficacy giving a 22 guidelines, and multidisciplinary having 11 guidelines. And for preparing the CTD, I was following the quality guidelines completely at 26 guidelines, in which especially I was following the ICH Q5A, Q5C, Q5D for the cell line preparation, cell line banking structures. I was using the ICH Q5A and Q5C for the stability data, and the Q5D for the characterization of the materials, and Q57 especially the GMP of usage of the viral vectors and restore or quality aspects. And the modules we'll be using for the CTDs, or the module 1 is the administrative usage, especially the prescription usage covering letter, administrative, legal documents, a brief introduction of the product. Module 2 is especially meant for the summaries and quality overall summaries, including quality. 2.3 of module 3 is quality overall summaries. In 2.6 we mentioned the quality non-clinical summary, and 2.7 we mentioned the clinical summary derived from the module 4 and 5. And module 3 is the important thing we follow from the reference of M4Q. In 3.2 yes, we produce we document the drug substance material, and 3.2 P is the drug product documentation or drafting material of drug product, includes the extractable leachables and the final control strategies. And module 4 is the non-clinical study protocols as well as reports in this from the animal studies as well, preclinical studies. And module 5 is clinical studies, even the clinical protocol, drafted protocol as well as reports. We especially use the references for these CTDs from the quality M4Q referral to document. For module non-clinical, we use the M4S for module for safety report to, and module 5 clinical reports we use the module 5E. Module 5E reports and course efficacy. So yes, these are the terms and scale sets I have understood from the course which I got from the Fortunate Veda. Thank you.
Yeah. Is it end from your end, Partha?
Yeah, it is end.
Okay.
14
Okay.
slides I
So
have
before
prepared.
going to Q&A. So I will introduce some other topic, so which is necessary to understand, especially for the cell and gene therapy. Correct?
Yeah, sure, 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 sure.
Yeah. Can you stop for a while your presentation?
Done, sir. Done.
Yeah.
Yeah.
Yeah, I'm coming.
Mm.
Yeah, I'm saying, so which is the intended purpose, we should want to know that how to the cell and gene therapy, what are the suggested that. So in especially for the US filing, so where it is filing and exactly. So if you see
Yeah.
in this slide, regulatory centers for FDA and Office of Medicinal Products and Tobacco, just leave it the tobacco terminology. Office of Medical Products, under there is a three parts. So one is a CDER and your CBER, and there is a
Mm.
CDRH. So one is for Center for Drug Evaluation and Research. Under this, there is a Office of Therapeutic and Research and Review. So under this department, there are divided into two departments. One is for chemical drugs, which is generics, and as well chemical drugs. And coming to another department, Office of Biotechnology, which will talk about therapeutic biologics such as MAPs, growth factors, enzymes. Enzymes is we can call it insulins also. Insulin and analogs will be fall under this, and any immunomodulars, so which will fall under CDR branch. Now coming to second branch, Center for Biologic Evolution and Research, there is divided into three departments. So one is OBRR, nothing but Office of Blood Research and Review, related to blood products. So we are going to file for blood products. We should approach to this path and followed with this
Okay.
path and followed
Mm.
with this path. Now coming
Okay.
to cell and gene therapy. So coming to especially the topic which we discussed so far. So cell and gene therapy, when want to file in the USFDA, so enter into Office of Medical Products, and there is a CBER. From under that, there is a OTAT, is nothing but Office of Tissue and Advanced Therapies. Tissue and Advanced Therapies. Under that, we have to file cell and gene therapy products. So it will fall under this pathway for entering into US filing. And one more, OVRR, Office of Vaccines Research and Review. So vaccine is having separate pathway. So that very clear. So therapeutics is having separate path, and blood products having separate path, cell and gene therapy having separate path, and vaccines having separate path. So which is the third one? So which is having a Center for Devices and Radiological Health. So this will fall under medical devices. So anyhow, it will cover in ongoing batches. So just I want to tell that since we are talking about cell and gene therapy, this is the path to enter into FDA market. Yeah, is it clear to everyone?
Sir, we are losing.
Sorry, can you repeat again?
Like I was losing you. I don't know whether in between your voice was breaking.
Is it to all, or it is for especially for Imanshu? Is it clear to everyone?
It is clear from my end. Actually, I can hear you. Exactly.
Yeah, what about others? Nassim, are we clear?
Yes, sir.
Okay, Imanshu, I think so from your end it is
Yeah, I
working.
maybe like in my
No
end
worry,
it is unstable.
no worry. See, when you want to file cell and gene therapy product into US, so we should know the pathways. Correct? As a regulatory persons, we should know the pathway. So when entering into US FDA site, so there is a we should enter into Center for Biologics and Evaluation Research. Under there are three departments. From that, we had to reach to Office and Tissue and Advanced Therapy department. There we had to put our application for to file cell and gene therapy products.
Mm.
This is what I
Okay.
want to tell it. So which is missing in
Yeah. Correct.
Partha's session. Yeah.
Yeah. Yeah. Yeah.
Got it,
Okay.
Sir,
Himanshu?
one more thing I want to highlight
Hmm,
here,
hmm.
like biologics are a kind of like it should go into the Center for Biologics Evaluation and Research, but how come these are into this CDER? This is just a drug evaluation and
Again,
research.
it is divided into two parts: chemical drugs separately and Office of Biotechnology Products is separately.
Okay, but
Yeah.
there might like the confusion might happen that it is biologics. Why it is not under the CBER
Uh, uh,
Your question is well valid, but the branches they're
under
already
the
categorized
biologics?
like this.
Is there any
mm,
background associated with it?
I need to verify. I'm not sure, but as per the design, the pathways which is already created. Actually, what happened? The filing products, there are pathways like 501 and 351. So for chemical drugs, we'll go through 501 pathway, and for new biologics, we'll go through 351. There is a 351A and
A,
351J.
K,
So
A, and K.
okay, correct. So that pathway we have to enter into. So that is what previously we discussed in previous session. I will clarify in our session because I'm not prepared for that. I will come back. That is a good question, but somewhere it should be mentioned regarding that filing pathways. We'll come back for that.
Okay.
Hmm. Just I want to clarify since it is a missing information. That's what I want to tell you here.
Yeah,
And that's
Yeah.
true.
that's very good, sir. Actually, the reason I highlighted this here
Mm-hmm.
because there is one department called CDER, that is a drug evaluation department, and
Mm-hmm.
under which we are evaluating the biologics material. Similarly, they have also made a different wing called CMC, right? Under which we are having the blood products and this gene therapy and all those things. But
Mm-hmm.
biologics are not there. Ideally, biologics should be there.
Yeah,
So
I'll clarify that one. So since
yeah.
it should be fall actually if you speak what are the question you ask, these two falling in one file pathway. So which is CBAR and CDR. So under that there is a chemical drugs which is 505B and 505J pathway for new
Mhm.
drugs and as well as generics, and for biologics 351A and 351K for biosimilars and for new biologics. So regarding whatever you ask the question, it is as of now no answer for it, but I will refer and I'll update you. Yeah.
Okay.
Mm.
Thank you for
Yeah.
enlightening this presentation today.
Yeah, fine,
I
fine.
mean,
Not a
I'm not aware of those pathways, especially for
no
submitting.
worry, because that is falling under
Yeah,
operations filing
correct,
pathways.
correct.
So as
Yeah.
a CMC point of view, we should know that
Mm,
as per model 3 requirement, whatever you presented, that is model 3 requirement. The
correct.
prepared model
CMC.
3, the prepared
Mhm.
model 3 how to file into US. So that is the path to have to enter into that path.
Okay. Okay.
Yeah. Yeah. So any questions from any participants, we can discuss. So we have another almost 15 minutes time with us. So we can go for in detail for the cell and gene therapy related particular topic.
So Pardhasaradhi, I just want to understand few more things. Like for the characterization, what are
Mm.
the techniques they use? Can you shed some light on this?
Yeah, in the case of characterizing of these viral vectors, we are, especially as per regulatory norms, we have to use a flow cytometry evaluation and flow cytometry, digital droplet PCR, LCMS, and dynamic light scattering. These are the main four equipments we'll be using for the characterizing of the molecule of our product. So in the flow cytometry, we have to, in the case of lentivirus, we'll be using a flow cytometry. We'll be treating the cells using transduction of the virus over the cells, and the cells will be pumped or sampled to the flow cytometry, where we, with the help of a specific detector over to the cells, it's a kind of marker, I would say. It's a probe in a way. The probe will detect the specific marker on the cells that yes, cells got infected with the lentivirus. So hence the potency will be evaluated from there. So that is a critical thing for the characterization of our raw material of lenti. Since again, in case of AAV, we'll be having a in vitro potency studies over various cell lines. And in case of dynamic light scattering, so at specific nanometers, we'll be studying, understanding the size distribution there. So that is also another characterization of the viruses, where we'll understand the full and empty or partial viruses will be differentiated there. And obviously, in case of another analytical distribution, we have to do is that is with the ELISA. In the ELISA advanced ELA therapy, ELA will be machine will be evaluated there, where with the virus sample we'll understand the amount of partial or a full or a empty full capsids will be evaluated there. Yes, these are the five things
And
we have to
are we
try.
using LCMS here for sequencing, or it is like a different kind of sequencing method? Because these are
in
the nucleotide
in
sequences.
yeah, in LC we'll be understanding various type of LCs are there. In LC we'll be finding the amount serotype, especially not the gene of interest inside the sequencing, but the serotype, which serotype we are getting from the product, whether the serotype is not contaminated with the other serotypes. These things will be looking at with the using LCMS.
These are the sequencing only, right?
No, no, you will get the peak signals there in LCMS. We have purchased it. We have not yet evaluated at the moment. We will be doing that very soon. But yeah, as per the literature, what I understand is this: we'll be getting a specific signals with the viral particle genes in it. Accordingly, VP1, VP2, VP3 signals will be generating in the chromatogram there.
Okay.
And in case of sequencing, yes, there will be another third party will be evaluating. In sequencing of the gene of interest, we'll be doing that. These are the characterization, especially characterizing the equipment, characterizing the machines we use.
Okay, so is there any orthogonal method of analysis for the characterization, or we are going with a single method of analysis?
Usually, in case of plasmids, we use HPLC as well as gel electrophoresis for the supercoiling of the plasmid quality, which is a critical thing, which is a CQA in a way. But obviously, following the principles of QBD, we'll be using the method development. And yes, in case of plasmids, is that in case of viral vectors of raw materials, we'll be using the methods which are approved by the regulatory norms.
Okay, so cell and gene therapy is very new and it is advanced therapy. So
Mm.
as of now, what are the
Yeah.
products approved in the FDA or EMA or any known products names?
Yeah, there are many products in the market now, especially Zolgensma, which is a serotype AAV9. Chimera is one of the CAR-T system or
CAR-T
CAR-T.
cells.
Yeah, there are
Hmm.
other CAR-T products in the market as well, related to various even FDA in US as well as in UK, EMA as well as got approvals. And Glibera is one of the product, which are renowned product, which I'm saying is a very renowned products. And there are many products in the pipeline. Many products got approved as well.
Okay,
Mm.
so especially the cell and gene therapy products, is it single dose or it will be multi-dose? How it will be?
It depends on again disease and serotype, especially in case of few diseases there are only single dose with a long lasting advantage with the single dose with the product. And there are few products with again two doses. It
Okay,
depends on patients as well as it depends on the disease.
so what is the time duration for the between doses?
With to my knowledge it is 1 month to two
Okay,
months
30 days. Okay, fine. Yeah,
Yeah.
okay. Hmm.
Is there any formulation aspect associated with these kind of product? Means, and
Yeah,
if
as I
it is,
said
then what
in
is so?
yeah, as I said in the formulation, pluronic F68 along with the glycerol, which is very essential part for the stability at minus 80 degrees centigrades. So along with that, we also use sucrose to stabilize the capsids. So yes, these are the things we special for the
Okay,
stability.
formulated drug product, it is necessary or it is stable at even 2 to 8 or not stable at
No,
2
no,
to 8?
it is essential to formulate and to ensure to store at minus 80s.
So what is the thawing kind of thing prior to taking into patient's body?
So we thaw it for repeated thawings, the virus gets instabilized. Because of
Correct.
that, we use always glycerol and pluronic F68. So that will make ensure the repeated thawings to make the shear stress damage. So to in order to mitigate that, this glycerol helps that.
Okay, so
So,
that
yeah.
vial is a single use vial or it is a multi-dose vial?
I'm not sure. I'm not into that domain,
Correct,
but
correct. So why I'm
yeah,
asking, it is a single use vial, we can thaw it and we can use it for the
correct,
patient.
correct.
It is a multi-dose vial means we had to thaw it and use it and remaining quantity had to reserve for next patient.
Probably it will be single use, probably because
Oh,
it I never heard about multi dose in one
okay,
vial.
okay, okay, great. Yeah.
So
So
as you said, yes, sir. Yeah, you can go ahead.
proceed. I will come
Yeah,
up
I
last.
have one question.
Mm.
Because as these products are stored in minus 80,
Yes.
so generally, if you see, like the we are still having the challenges in cold chain. Okay.
Correct.
So like in the like when it goes to the market, like
Mm,
what about the cold chain things? How we are ensuring that there should not be any kind of issues in terms of stability, because cold chain things is still the things, like how we are ensuring that this is very much safe and effective
yeah,
in
true,
those
true,
point of
true,
view, supply
true.
chain point of view.
Yeah, correct. So, yeah, keeping in that point, especially we even do generate more designing of the plasmids, more rapid plasmid, more chimeric plasmids in order to sustain to the temperature aberrations or the stability aspects. So, still process is going on. The development is a never-ending story, as you know all. But to my knowledge, until now, the process of product which are in the market are having a very temperature supply chain management, which are having a good understanding and provide with the risk assessments and gap analysis. They have a supply chains with a strong supply chain of the cold supply vendors.
And may I know what is the accelerated stability of these products? Stability.
Since it is minus 80 product, acceleration will be below 5 plus and minus 5 degrees centigrade.
Okay.
Today, nothing but 2 to 8 degree. 2 to 8 degree is nothing but.
Yeah, to do it in a way.
Yeah. Any questions from others? The people who are late join, will share the recorded session so that you can go through it. Bhushan, do you have any questions?
Actually, I just wanted to know,
Mm,
in case of ELVs, this is apart from some regulatory aspect.
mm,
Since I was part of downstream processing, we
mm,
know that ELVs are a little in size larger than AVs and more shear sensitive or
mm,
fragile molecules.
mm.
So, is there any particular uh, care or precautions we need to take to intact its functionality, or what we used to do its potency functional titer?
In case of lenti, you're saying? In case of lenti downstream?
Yes, lenti downstream.
In lenti downstream, we usually do. We have got a very good amount of potency, recent studies. So, in our process, we especially do. First step is to do a TFF. So, in the TFF, where we have seen a loss in the material there, so we have evaluated various TFF filters in recent studies. And further after that, we have done a capture step in the chromatography. Then again we have done a TFF and diafiltration.
Okay.
So in this process we haven't seen any major losses, and especially lenti clarification we have seen a loss, and we are doing a study on that. And yeah, there was no damage at the time of potency till now.
That's
Mm,
usually during TFF operation, some shearing
mm,
stresses increases, which leads
correct,
to
correct.
loss its functionality. That's why
Mm,
my question.
mm.
And apart from
So
that,
mm, so since we have made a clarification, I mean TFF study in house with a very slow flow rates, we haven't saw any aberration in the potency currently. But yeah, it's a very good point. We will evaluate in that case with flow rate differences.
okay, and again related to that only,
Mm,
as we are more focused on functionality of molecules,
mm,
in the samples which are
mm,
having less concentration, like in case of TFF, we say permeate, or in case of chromatography, whatever
correct.
flow through or washes will be there.
Mm, mm,
How
mm.
we use, how we can detect its functionality, since its concentration will be very less.
So we always collect in process samples from the main feed material, then the permeate material, as well as the retentate material at various different concentrations. We always calculate the concentration factors at various time points. So we collect those samples and do the potency studies. Initially, prior to going to the potential study, we go with the dynamic light scattering. I even recommend for you as well. So in the
Okay.
dynamic light scattering, if you see any signals, or a structural material nanometer signals there in
Hmm.
the permeate.
Hmm.
Make sure then you can further do a further recirculation.
Okay. Uh, at least qualitative, we can get
Yes,
information
correct,
whether it
correct.
is
Mm.
how much
So,
loss we think we can't.
correct. So
Mm.
this required to establish during our development by using DOI concept portion. So to handle the
Yeah.
TAF for the flow rate, what is the cross flow rate, and as well as for permeate flow rate, flux rate, and TMP, what TMP need to be applied. In general, in this,
Those part related to its operational, but
Mm.
in analytical aspect, if we check for
Yeah,
functionality tighter,
correct.
sensitivity will be required more compared to
Mm.
correct,
some
correct, required.
power
So
3, power 4, something will be required minimum as per my
yeah,
understanding.
correct. Since the productivity is high costly and sensitive
Mm.
operation, so we can put some sensors which is can be monitored, able to monitor
Mm.
the UV. So when you are
Ye-
especially the virus particles to be monitored through UV like 260, 260 by 280, some 320 will put it into monitoring.
yeah,
The ratio of OD based on the OD, we can able to control if anything is moving from redundant to towards the permeate side, any loss is there, we can easily can detect it. So there is a permeate flow rate permeate line will put it UV monitoring cell, as well as in redundant also will put it, so that we can able to monitor any losses as well as concentration of the viral particles.
so Bhushan, you work on lentivirus.
No,
Yeah,
I worked on Sputnik vaccine.
Okay. I
Uh,
mean, in the case of lenti, I would recommend to do ELISA. Uh,
yes,
since
uh,
you cannot do
ELISA.
all at a time a potency study, but you can do a ELISA study to
Huh.
evaluate the written date permeate samples. As I said, you
okay,
can go for a P24 assay.
correct,
Huh.
correct. So if you want a specific
So we
point
do
of
that
view
P24 assay.
selection, ELISA method is the right
Mm.
kit method to use. But I'm
Yeah,
telling as a point
correct.
of view, quantity to control, as a quality,
Control, yeah.
as
Yeah,
a
your
Huh.
point,
quantity
right? Mm.
control.
As you mentioned,
These people,
P24 assay is also
yeah,
one of the solution if we check in this case.
Correct.
But what happens is whatever in permit free P24
Mm.
molecules will be there, those will
Mm.
also get analyzed. So whether those are intact LVs which we are losing there or free P24
Yeah,
molecules, that is
if
also.
in that case, yeah, I would also recommend if you have that queries. I mean, if your material is leak into retentate, I mean
uh,
permeate, then you can do a qPCR, which is LTR specific using LTR specific primers.
Yes, yes.
You can evaluate that. We have done that in our lab.
And again, related to this only,
Mm-hmm.
in case of impurities, means apart from, in
Mm.
case of PV again,
Mm.
apart from free P24 molecules or SCD, SCP, which we use to check, any
Mm.
other impurities process related or product related, you guys have observed anything or any
Um,
checks? As such?
right now, in case of release criteria, we were using the kanamycin release
Okay.
criteria and plasmid, some plasmid promoters. We'll be using them. Apart from that, no other impurity strategies. We are, of course, single the plasmids, supercoiled, those things we will be evaluating. But apart from that, in case of lenti release criteria, no other. P24, your P24 LTR specific qPCR, then your potency. Ideally, the potency using flow
Ideally,
cytometry.
potential. Okay, right.
Flow cytometry data is needed in regulatory.
Uh,
And now, yesterday, there was a guideline released for the flow cytometry stating that there should be no variations between the sample. It was so stringent,
which is very
very
challenging.
stringent, very stringent.
Okay. And in case of AAV,
Mm,
I said to,
mm.
which we used to check ratio of
Correct. Full
empty,
and empty ratios.
empty full, and again
Mm.
sometimes partially filled or
Correct.
overfilled
Correct.
particles. So,
Correct.
is there any guideline or any final release
Mm,
check it is there
mm.
or range for this ratio?
No, until now there is no guideline. We have a good consultation from Paraxel regulatory consultants as well. In the last conference meeting as well, I have a similar question as well. I was posting to them. He was saying there was no guideline until now with in respects to the full and empty capsids until now in the release criteria as well. Whatever you are getting at the end of final, you can mention the CTD,
Okay,
but
so
ensure with
means
a good quality. That is the main potency, right?
means no chances of failing of batch or sample kind of there.
Yeah, yeah,
Whatever they
yeah.
show,
Mm.
got it. Apart from this, any other releasing criteria are there for AV?
For AAV, as I said in my presentation, there are many release criteria, such as uh, mm, replication competent assay is one of the critical thing,
Yes.
and ITR specific gene sequencing is another criteria, and the AAV capsid identity of the serotype is another criteria, and obviously empty and full ratios,
Full ratio.
and DPCR is another one which is essential nowadays to release, and
One more
potency.
thing which I have observed or we have faced that in case of where we are using multi caps or um, uh, like multi angle
Light
light
scattering.
scattering.
Right.
Okay.
In that cases,
Mhm.
if we try to separate the partial field and field empty, these three capsids, now the
Mm,
resolution is there
mm.
not that up to the mark, so we can say separate those. Have you
Yes.
observed them, or your guys are able to?
Uh, uh, since I already said we have just purchased our LC, and we will be evaluating a study on the full and empty capsids and that. But until now, to my knowledge, the way we process is like we purify the empty capsids in our second step. That is a polishing step in the downstream.
Hmm,
Then we evaluate a full and empty capsids there in the ELISA, ELA, for
okay.
along with the DPCR data. So there we correlate the full and empty capsids, and we see a drastic difference from the upstream process to the final step
Yes.
of downstream step, second step of downstream.
Okay. And for separation of full and empty, we are going with AEX or density basis separation you have.
AEX density
AEX.
based. I don't feel it's a scalable thing, but we
Not
are going with
scalable, but
AEX.
until now it was the only proven. So
Mm.
traditional method we can say in
Yeah,
this case.
traditionally, density gradient we can generate a in-house reference material, I would say,
Uh, for
and
small scale, okay.
small, yeah, small scales, but for the large scale or a scalable way, I would
AX is only
prefer
alternative.
AX. Yeah, I have seen publications and people are getting at 90% purity with full capsid.
Uh, on publication or papers, we are seeing the
No, no,
numbers.
even
But,
even case of industry as well, I have seen 90% of full
oh,
capsids
great.
in the
Okay.
final
Might be
purification.
optimization required from our side.
Yeah, yeah, much optimization. It's a challenging
Uh,
step in the downstream.
yes, yes. Okay. Nice to meet you.
Where do you work, Bhushan?
Now I am associated with Biocon, but last of my organization is Intas, where I was
In
part of.
TAS. Oh, okay, okay, okay. Yeah, thank you.
Okay. Thank you, Bhushan, for interacting nicely. And is there any questions from any others? Narsama, do you have any further question? Dr. Narsama?
No, sir. Thank you. Nice presentation, Partha. It's
Thank you. I'm
I
glad.
admit I'm new to the this virus and all that.
No
So
worries, no worries.
great.
Thank you. I'm glad
Yeah,
to hear that. Thank you.
thank you.
Okay. Okay. Iman, should you have any further questions?
No, as of now, I don't have any question.
Okay. Okay. Great. Thank you.
Thank
Thank you for
you.
attending and
Thank you.
spending time with us.
Mm.
Pratap?
No questions, my side. Thank you, sir, Partha.
Thank you, Pratap. Thank you very much.
Okay. Priya Soni?
No any questions.
Thank you, Priya.
So, always you are understanding cell and gene therapy pathways? Okay. Okay. Fine. Usharani?
No, sir, I don't have any questions.
Thank you, Sharani.
Okay. Okay. So, I'm stopping the recording. After the recording, also, if anything, we'll clarify it. Yeah. Thank you for joining and sparing time on weekends.
Thank you, sir, arranging for this.
Thank you, organizing,
Yeah.
especially.
Yeah. It's